Okay, why don't we get started then? Gilberto, take it away.
Okay. Uh, well, let's continue with our cell properties and behaviors. Now, I'm going to introduce you to how to make a cell growth and how to make a cell suffer mitosis to divide itself. Um, let's go there. This is the outline of this lecture. We will introduce some motivation about the cell division and cell growth. Then I will implement some exercise using the tools that CompCell3D offers to us to implement those biological processes in the simulations. Okay, this is three exercise. Uh, I will put in the screen the exercise itself, and I will go over it, and then I will code it on the screen with you. Okay. Okay. Cell proliferation division. This is the basic stuff that creates a multicellular organism. You know? The cells must all the time decide when they have to proliferate, so how to they have to survive, how to have differentiate to different organs and make a complete individual, or even to die. Huh? This kind of processes are very complicated and are based in intricate range of internal external signals with some proteins regulations, lots of biochemical process. Um, a bunch of these factors that decide that constitute a cell in a given time is that we call the cell state. Okay, a cell state where all these processes are activated, and we don't know for sure which ones we it's we don't have much knowledge about everything of this process yet, but something we know for sure. The principle of this process for division is the cell cycle. When the cell decides to proliferate, I mean to divide itself, it enters in what we call cell cycle. This cycle has four phases, which we can see there: prophase, metaphase, anaphase, and telophase. Sorry for my pronunciations, but anyway. And this cycle is compound by very four stages, which we call M, G1, S, G2, and G0 if the cell stops to that decision to start to proliferate. So this is basically what the cell does to proliferate and multiplicate itself. Um, there are some checkpoints in this process where the cell decides to enter or to get out of some phase of the cycle. There is a variety of controls that uh, make the cells follow this cycle. And if the cells, if the checkpoints are fail to to produce the cell cycle, the cell cycle stops and the cell stops to proliferate and often they go to apoptosis and die. There are lots of questions, let's say, if the cell has to make for the cell cycle continuous, okay? But anyway, we don't pretend to be that that specific in our model. It's too complicated to do that. Huh? This entire cell cycle is not our scope today. We are going to take a one cell and see how we can create some tools, mechanisms that we enforce the division during the simulation. Of course, if you a cell doing that, it it needs to know if there is available space in the medium or where the cell is, if there is amount of nutrients taken up is enough, how fast the nutrients can be transformed into cell mass because the cell has to double its size before it divides, and lots of process a cell has to follow before get one division, let's say. Let's start with a simple rule. Our cell will grow, we will divide, and we will divide. Just like that. We will impose a rule for the cell to divide. Okay. Okay, how we do that in CompuCell? We saw previously today that when you create a simulation that will include the plugin volume, that volume plugin came with two parameters by default, defined by default, or you can change it. The two parameters are the target volume and the lambda volume. We saw that there. But we can impose a changing in the target volume during the simulation by saying that each amount of time that you will define how big it should be, this amount of time, the target volume should increase like that by 0.001 1/10 of a pixel per Monte Carlo step, for example. I mean, that means that a cell will increase 1 pixel per 100 Monte Carlo steps. Okay. Okay. So this is a simple rule for growth. You add something to the target volume in an amount of time, in interval of times you want. Okay. Let's do the following exercise. I'm gonna go over it. So we are going to build a simulation with 100 by 100 points in the grid. We'll create a single cell and assign to it its contact energies and initial geometry. I mean, it's what we have done so far in the basic simulation we did in the previous module. We are going to use the blob initializer this time, not the uniform initializer, to create a rounded cell in the center of the simulation domain. We are going to save the project. Now we add the plugins contact, volume local flags, surface local flags, and the center of mass plugin. The same as we did before. But now we are going to add the steppable growth. How do you do that? And I will show you later. Continue, we edit the blob initializer to say that the cells will have a radius 5 voxels or pixels. We will edit the start function, and that's where we have to make a little extra slide to explain how the Steppable works. Okay, the Steppable works. We is the place where we build our own functions that the cell has to do. Or if you want to measure, if to want to plot output data, if to save data in your hard drive, anything is doing this in these steppables. I'm gonna tell you how to do that. In the steppable, we'll give to the cell the initial target volume, and the lambda volume equals to 3. In this step, the steppable has many functions that I will show you later. And then we will edit the growth steppable, that is the one we include in the wizard. This growth steppable will say to the cell how it must do, how the cell must grow. This is the function we just have seen in the previous slide. Okay, let's build that. I will let the exercise here, and I will start code here. Okay, so create a new project here. You can give it the name cell growth if you want. Again, every exercise is in the student materials. If you don't want to do that, you can download it from there. Cell growth. Next. 100 square, blah, 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 no, block. Okay, we have one cell. That's cell. Add. No chemical fields. Contact. Volume. Center of mass. And growth here. According with our slides. So contact, volume flux. I forgot the surface local flux. And the growth. Next, finish. Okay, here is the simulation, double click, then you have the file is open. See, I'm going to the XML script to format the blob where the cell is being created here. See, blob initializer project, steppable. Radius equals to 5. 5 equals, both equal. Then we are now it's the time to talk about the steppable. Look, let's go to the slide again. The steppable then is the part of the simulation project, the file where you allow users are allowed to build anything they want during to be executed during the simulation. In general, steppable has these five functions here. Okay, and they are called init, which is setups. This is the corrector from PowerPoint. Should be steppable here. Set up the steppable frequency call, which comes in here or here. it says how often your steppable is called from your main Python file. You have the start function here, where this start function is called at Monte Carlo step 0. When you click, if you start your simulation with the step button there, the only thing that simulation will do is to initialize that start function there in the step-up. Nothing else. No, not only one calculation. Okay. So everything that has to be initialized in the simulation must be put in the start function. The step function for its time, it is called at any other Monte Carlo step after, accordingly with the frequency you call your steppable. If you call each 10 Monte Carlo step, this step will be activated each 10 Monte Carlo step. Okay, but we have here at each Monte Carlo step one frequency. So in this step function, you put anything you want to calculate, update, plot, save, anything. Every calculation you want to do and want your cell to do come in this part of the step, in the step function. Okay. When the simulation stops, it goes to the finish function. And here you can put something to print on a screen or give you a message, say, the simulation has ended, something like that. In a good way, for example. And the on-stop function, if it's a function that is called if the simulation stops before it finishes. And also you can put some message here to say something about the why the simulation stopped, or if some condition was achieved during the calculations, something, anything. Okay, so this is the structure of the steppable, and this is very important because you can build lots of things here. Remember again, I will remember again that from simulation I showed the last one, the complex simulation I showed yesterday with that bunch of plot windows, which are plotting many calculations, statistical calculations. All that calculations are made inside the steppables. And actually, you don't need to have only one steppable. A steppable is a class. You can have many classes as you need for your simulation, and they can be called at the different frequencies. Okay, so now let's go back to example here. So in the constraint initialize steppable, see this one, it comes with the simulation. It came with the simulation. It is a default. It has its default values of 25 for the target of cell volume and 2 for the cell lambda volume. And see, it is inside a loop if in case your simulation has more than one cell. Our simulation will have only one cell at the beginning. Okay? So this loop is self-cell list. It's a list that it contains all the cells built in the growth here. When you initialize here, see, the width and the radius are the same, so you are only build one cell. If you have something different here, you can build lots of cells. If they put a radius 50, for example, you have a bunch of cells with radius 10. Okay? Let's go to the Steppable. Okay, so now look, I have the start function here with those values, and here I have the growth steppable, another class, as I told you before, which comes with a default in increment of 1 pixel per Monte Carlo step, which is too fast, growing too fast. So we are going to use this value here, 25 voxels per each 500 Monte Carlo step. So we'll edit this here and put 25 over 500. Okay? So after 500 Monte Carlo steps, we have a cell doubling its size. It will take 500 Monte Carlo steps to cell double its size. Okay? So what else? So this is what I have said already. Also here we have a loop for the cell list, because if you have more cells, all the cells will grow at the same rate. Okay. Now let's run the simulation. Save it here. And open in player. I will squeeze here the simulation. Where is my here? Where is my cell? Something, I missed something here. I know. I forgot to adjust this. I see. 3 for the lambda volume. Let's see if it works now. See, there is the cell. I hope. Yeah. It's too small. I don't know why. I forgot something. This is the problem of to code online, code in the. I didn't save it here. I forgot to save the simulation. It's a very usual problem. See, the cell is increasing with time, slowly, because it takes 500 Monte Carlo steps to double its size. And the cell starts very small. So now we have about 9,000 Monte Carlo steps. The cells have multiplied by 15 almost. Okay. Now let's follow this exercise. Now, on a spoiler alert, I'm going to introduce something that you probably will see in the next module, which is how to create a graphic output window. Okay, I will introduce this windows because I want to keep track of the growth, to see how the cell is growing with time in a graph. Okay, so let's do the following. In the growth steppable here, see, this growth steppable doesn't have a a start function. It doesn't need the start function. In principle, the growth alone, the growth steppable alone, doesn't need to be initialized. It starts when it starts is in the step function because it starts to increase in the target volume. So we needed to initialize a graphic window. To do that, I have to create a start function. Tab start self. Where is it? Here. And in this start function, I will create the plot window. And now I'm gonna use that tool I talked in the end of previous lecture, which is the snippet here from CC3D Python coding, a tool called scientific plots. In the scientific plots tab, subtab, actually, I will add the snippet which belongs to the start function here. Understood? So I will click here, and this block appears. I will what is in this block? See, it creates a plot window with the name plot underscore window. This is the variable which contains the object window. The name of the variable is plot window. Self is because you can use this for the whole class. It has some parts you have to edit. For example, the title of the window. So I'm not measuring the surface, only the volume. So average volume or cell volume because I only have one cell. The X title, Monte Carlo step. The Y title, volume. Here we have also the scale of how do you want to your axis behaves. Linear scale, it can be logarithmic scale, but the logarithmic scale, I think it's bugged so far. Don't use the log scale until new version is released. And you can say if you want to show or not the grid of your graph. And then you create a placeholder for the curve you want to put in that window. Okay, so here is the window. And here you create, I will comment this line here. We'll create the placeholder for the graph itself. So I will put here with plot with a line which had a core red and with 5 pixels. So I am defining the format of the curve. It should be plot with lines, red lines, size 5. Okay, this is how the graph is created. So again, it is in start function at the beginning of the simulation. If you click only the step button, a black window will open together with the cell window. And after that, I have to say in the step function that I want to see the cell volume plotted in that graph. So I come here inside the loop because I may have more than one cell. go back to the CC3D Python tab, go again to the scientific plots, and now I'm going to choose the add data points. See, these add data points goes to inside step function and click here. Okay. A new block was put in here. I don't see it by default it puts always two placeholders for each Windows window graph. But I'm going to use only one for now. Sorry. So I comment this one at the start. I will comment this one at the step. So, excuse me. Since I called the placeholder as vols, here I will call also a vol. And I'm saying this line self-plot win, I mean, inside the graph plot win, add a data point in the placeholder vol. Which is, what is this data point? Is the MCS and I cell target volume or cell volume. Let's put cell volume because I'm okay. That's good enough. So now we can see everything is described here. Both the snippets coming from the revising. You go to the tab CC3D project, CC3D Python tab. The drop-down menu go to scientific plots and the step and the add point. That's what we have done here. Okay, now we can run the simulation. One step. See, I will try to keep the slide together. I will click here. I will tile this graph. This is something has to do with the zoom stuff. This, if you put in outside the zoom, the axis will be corrected. So we have here, as I told you, I start the simulation with only the step button. I have the cell and I have the graph window. Now I'm gonna put it to run. Since we are updating the graph each Monte Carlo step, we have this big curve here. I don't know why something has happened with this.
Can I have a question?
Excuse me?
Can I have a question?
Yes.
In the cell volume graph, we have ups and downs. How does this occur? I mean, we just gain voxels and we never lose voxels, but still we have up and down.
No, you lose because the cell is growing, but the membrane fluctuation of the temperature makes the cell oscillates. I don't know why it's not. Just a second. Let me come back here.
Excuse me, Catherine.
Yeah.
We are increasing the target volume, not the volume itself. So
yes, the volume can change.
Okay.
See?
Okay. Thank
you. Something happens when I put the simulation here in the zoom. It's the cell window stops, freeze. It's like I have to keep clicking here. But you see, it works. We can see the growing here. Okay. Okay, let's continue then. This is what you should see. And this one I had the grid turned here. If you put the grid equals to true here, you have this image like you see in the slide. Okay. Now let's continue the implementation. Take into account the surface of the cell surface. Remember, when we load when we create the basic simulation, we include the plugin surface flex at the beginning. But the surface flex doesn't bring you in the start function the values for default values, any default values. It just was loaded inside the XML, like here, see, line 29, but no parameter set, like the one. We have to put the initial values here in the steppable. So let's do that. I will duplicate this line here. Oops. And I will change here to surface. And surface. And use 20. See, our cell at the beginning is a square, not a square, it's a blob of radius 5, but I'm defining its target as a square of size 5. And also with the same value for the lambda surface. Okay. I will add now a next placeholder for another graph, the surface graph. Here. You have also, let's put lines with another color, green. Okay. And I have to add another line in the step function to plot the instantaneous value for the cell surface. Is that okay? The same thing as I did before for the volume, I did now for the surface. And let's start again. Let's stop this one. Windows time. Again, I have empty graph windows. And when I let it run, I have to curse. The red one is for the volume. The green one is for the surface. And here, see, the cell doesn't evolve. Because it's very strange. Why isn't this zooms? Doesn't update the cell, the graphic 0 window here will show that the cell will go as a square. Because, you know, the cell, we are not increasing the cell surface, only the volume. The surface is fixed in that value of 20. We are not updating the cell surface yet. So it keeps pushing to the cell to be a square. And this is what you see here. See, the surface grows, but since it grows limited by the target, it grows much more slowly. The target doesn't change the surface target. Okay, so this is what the summary of the simulation. So let's fix this. Let's allow that the surface grows accordingly with the volume of the cell. How do we do that? We want to have this kind of situation here. The surface will grow, but grow according with the volume, and the cell shape should be round as it was at the beginning. And how do you do that? Let's do the following. Let's say here in the step function, I will define pi, the constant pi equals to 3.141596. That's enough, okay? And I would say that here, I will allow to the cell targeted surface grow. Oops. Surface will grow according. Well, since we are treating our cell as a circle, I will grow the surface as a perimeter of a circle. Okay, which is 2 pi times the radius of the cell. So this is going to be 2 pi times SQRT of cell dot volume. So now the volume is growing, but the surface is also allowed to grow according with the target volume. Okay, let's see what happens. Again, let's see if I can. It's very strange what happened there. Something is wrong here. I forgot something, sorry. Let's check. Actually, it was a good image I have there. Oops. What did I forget? 2 pi. Target part. Let's see, here's the graph. No, something is messed up. It's correct. 3 points, 2 points. Oh, I was duplicating the line and I duplicated it the wrong way. I mean, I'm not I'm growing the target, but not in a accumulated way. I'm saying in this line, in line 40, that the target is big enough to involve a circle with this target volume here. Okay, now it should work. No, I didn't save. Sure. That's correct. Okay. That's the image we have there. And the cell grows the usual way. Because I'm allowed with the surface plugin to the cell to adapt its target surface. Every time I use this function, it frees the cell. See? Okay. Let's continue here. Now we know how to do the cell, to make the cell growth. Now we have to start to trigger the division of the cell. Okay, so there is a small movie here that shows the complex mechanism for the division that comprehends that phases at the beginning of the lecture where the chromosomes are divided, pulled apart, the cell has to rebuild itself, to rebuild the membrane. It's a very, very complicated system. Have to recreate the nucleus and so forth. Of course, to do that is very, very ambitious. So we are going to do it in a simple way. Say we are going to divide the cell and it doubles its volume. We allow that to grow, keep growing during the simulation. When it doubles its volume, we divide the cell. So now we have to think about the problem. We have a division, so that means that one cell becomes two cells. We have to create a new one. Okay, because at the beginning we only have one. After division, we have two. A new cell is born inside the simulation. How do you call the cells? We call the original cell, the parent cell, we call parent cell. And the two remaining cells after division are the daughter cells. It's not a biological thing because I think it's in biology they call both they keep the name parent cell for the daughter. I don't. Anyway, so if you have a cell divided, which parts of the parent cell belongs to each daughter cell? That means how we divide the cell. Randomly, just say go half that, half here? Or should we choose a proliferation axis of division? CompCell offers you any possibility. You can divide the cell through the longer axis of it, or the smaller axis of it, or in a random axis of the cell. Okay, so we know how to divide, but now we have to say how do the daughter cells behave? What attributes are inherited from the parent to the daughters? Okay, it's a lot of information you need to give to the newborn cell, because here we have this new steppable that's called mitosis steppable. Okay, this mitosis steppable, it's a big one. It comes with these basic operations that I just described, like this choice of the axis of division, what cell, the attributes, how to update the attributes of the cell. But let's see the structure of this mitosis stepable. Of course, it comes with a default lines, and you have to adjust these lines the way you want for your simulation, adapt to your simulation. So, in general, a division is triggered by a particular event. What is this event? You have to choose. Okay. In our simulation, our event will be doubled its size. See, when the cell doubles its size, the division occurs. Okay? So the situation is that we have here the parent cell, the original cell, which is called self-parent cell in the CC3D. It's an object that exists before mitosis. Then you divide. It appears a newborn is a self-child cell. This is a new object, different from that one. So we have to give attributes to him. Here's what I was talking about. In our simulation, the parent cell keeps its characteristics. The daughter cell may inherit everything or not. It's up to us to decide how the daughter cell, the child cell, must be. So let's do this new exercise to create this, implement the mitosis steppable. Again, we'll start with a new project here. That I will call cell division. Pretty much equals to the previous one. 100 in a blob. I will create now two kinds of cell. ST, which stands for stem cells, and TD, it stands for terminally differentiated cell. I mean, a cell which is differentiated from the stem cell for any reason we want. No chemical fields, and now I will have contact. volume flex. I will not use this surface flex now, but I will load it. The growth steppable and the mitosis steppable. Okay. Don't need the neighbors. Is that it? Yeah. And finish. Then we have here. So let's go to the XML file here. And in the blob initializer, see, this is when, look, here's a difference between this simulation and the previous one. The previous one, I defined at the beginning only one type of cell, the cell that I called cell. Now I have two kinds of cells, ST and TD. So the blob initializer grab that information and try, if you don't do anything, he will create a circle with radius 20, and this circle he will create equally number of ST and TD cells with size 7. Okay? For example, if I write this, it will create 1/3 of ST cells and 2/3 of TD cells. This configuration can be done here in this very unique line here. But I don't want TD cells at the beginning because the TD cells are daughter cells. So I will start only with the only one cell equal to the previous simulation. Okay? Now I'm going to the steppables, and here I will use 25 and 5. See, start function in the class constraint initializer. Assign initial target volume to be 25 and lambda volume to be 5 for all cell types. Edit the step function here, and again use that growth rate of 25 over 500. Okay? So, let's save it. And here, I will Windows style. I will start this new. I have to, sorry, I need to open the simulation here in the browser, in the player. Open the player. No. Sorry for that, guys. I don't know why when I put the player in the Zoom, it doesn't work. See. The cells are dividing, keep dividing until fill up all the space. There is no control about the growing. The cells grow while they have space. But as soon as all the space is filled, they stop to grow. There is no more room to grow. They just keep wiggling the time, they trying to adapt, but nothing else happened. Not even sorting because the cells have same contact energy between them. Okay? When this situation is reached, we say that the cells are under pressure. And pressure is the difference between the target volume, the target volume and the real volume. Pressure is calculated from a force. Okay, now a little bit of physics here. The strength of a force acting in a system can be derived for energy, potential energy, like that. Force is the minus the gradient of energy. So when you apply pressure over something, this pressure is calculated like these ratios. Is the applied force over the area where it is applied. Huh? But force is the derivative of the energy. Which energy? The volume energy. Here, see? Is the energy over gradient of the volume energy divided by the area. This denominator here, area times length, it's volume. So pressure is the is the derivative of the volume energy in function of the volume. T dV. We know how is our expression for the volume? Is that one? Remember, I told you previous lecture. The energy term in the simulation is lambda v actual v minus v target square. So if we derive this expression here in function of volume, we have this expression for pressure. Okay? So we can calculate the pressure the cells are using this expression here. Let's calculate that. But before, let's do the following. We have this growing law for the simulation here in line 25 of the Steppable here. V target plus a constant factor, which is the growth rate. But now we can adjust this growing law, this growing rate here, saying that this growing rate is a function of the pressure of the cell. I mean, each one of the cells in the simulation here is under a pressure. So I will adjust the growth rate to this pressure for itself. Okay? There are many functions that I can do that. For example, I can adjust a linear decreasing of the growth rate. See, the G prime is a constant factor which multiplies a function of P, of the pressure. Okay? If the function is a curve with negative derivative here, the growth will be smaller the bigger the pressure is. Another possibility is here like a Michaelis-Menten function, like that. Or how do you call this? I don't remember the name of this function. But you can adjust the pressure also with this like a sigmoidal probability function here to act over the growth, decreasing the pressure of the whole cell, or at least to make them constant. Let's see how you do that. So now we rebuild this project here. I will not rebuild it. He's done here already. And I will introduce here in the step 2 of the simulation, I will introduce a cell dictionary called pressure. I mean, each cell has its own dictionary, and each cell, one of the features of this dictionary is the pressure of the cell. Okay, let's do that here. How you do that? Let's try to see the graph in a graph how the cell the pressure behaves. Okay. Let's start in the start function of the growth steppable, which doesn't exist as before. I will create this a graph window. Dept start self. As we did before, from CC3D Python tab, we include the plot. Okay. And this plot I will call how to say, AV TG target volume. Name of the variable is ABTGV. Here is AVval. And this one we comment. And in the step function, I have to plot it here. Let's include the line. Here, only one line. It's in AVval. So A capital A. And cell all. Look, here now it's time to do some calculations. As I told you before, I would stretch this a little bit. Look, now we don't have only one cell anymore. Because the cells are divided, the number of cells are increasing, so we have to calculate this average volume here. And how we do that? See, I will create here variable called vol equals to 0 at the beginning. And another parameter here, L, is the number of cells that I have in my simulation at this Monte Carlo step. Do you understand those two lines? This is a initial variable, average volume, and this L is the content. The length is the number of elements in cell list. So it's the number of the cells in the simulation at this Monte Carlo step. Now it makes sense the loop over cell list. Okay, in this loop now, I will calculate not the cell target volume, where is that in my calculation here? Okay, it's another loop here. Sorry. Here I will include the same cell loop here. Control copy, Control V, and I will calculate the average. You understand this loop? I am summing inside the variable avol the cell target volume for each cell divided by the number of the cells. So avol is the average size of the target volume of the cell. Okay? And I will add it, not here, because, but here. After I finish the loop that I'll calculate the average volume, I will plot this average volume in the window, the plot window, placeholder avol that I created here. Here is capital. Okay? Now I can. The growth is still continues the same as before. I didn't implement the pressure yet. I'm just looking what happens when I run this simulation. Let's see. Look. I will not mess with the graph to not froze anything. See, the average target is still grows, but nothing happens in the simulation anymore. This is only to show that the target continues growing, but there is no space, no room to the cell growth, nor divide, and the simulation is stuck, actually. Okay? So, now we can implement this stuff. But let us implement this contact inhibition growth. Something is wrong. Just a second. I think that are some. Okay. Oh, just a second. I think some slides are one missing here. Here. I don't know. Right. Okay. I was here. Okay. That's what you have seen there. Now we can add that this dictionary entry in the code to keep track of the cell individual cell pressure. Let's do that. So here at the beginning in the constant initializer, in the start function, remember the start function is the function that runs at the Monte Carlo step 0. We will add this new entry in the dictionary called cell.dict. Pressure equals to 0. Okay? And we have to include in the growth steppable. Let me a new line to measure and to plot and measure the pressure. We didn't do this yet. So a new graph window must be included here. Again, give it a space, CC3D Python. Look, it's equal to the previous one. I have to differentiate. How I do differentiate both? Here. Plot window 2. The second plot window. Huh? This plot window will measure the pressure. Monte Carlo step. Variables A, V, press. And here, again, only one window. And we'll call that A-V press. Lines, let's change the color just for change. Blue. So this is another plot window which is going to take care of the cell average pressure. We have to include here also about now in the steppable. We are going to run this. I need to plot. I have to calculate it before. Sorry. Here. At the same loop, I calculated the average volume. I will calculate the upper. Let's see. Update cell. Cell dict. Pressure. It's equal to that derivative we calculated last time. Oops, I erased here something. Okay. Remember that equation? Where is the equation? I want to show you the equation here. This one. Cell pressure is measured this way. So at the dictionary, I will put 2 times cell dot lambda volume times cell on what target volume minus cell dot volume. Let's go back. It's this line here. Cell lambda volume times cell target volume minus cell dot volume. And increment the average value. And now I can add a plot window. Oops. Add point to the plot window 2. This here. I forgot to put 2 here. At the window the press pre. Okay? So, resuming, in summary. At line 45, I define two variables to placeholder for the average values of volume and pressure. At line 46, L is the number of cells in this simulation at this Monte Carlo step here. In the loop in line 47, I have this counting the summing the cell target volumes and the average then by L, and summing the average pressure and averaged by L as well. Got it? And in lines 52 and 53, I am plotting these curves in the proper windows that I created in the start function. Okay, let's see. Okay, the cell are growing. The average target is grow to the double, the cell divided, grow to the double, divide, grow to the so far. And the pressure is basically constant here. I'm not going to press the tile button to not mess up with the simulation. It seems to be working fine. I will keep this running for a while just to see what happens when the cells fill the space. See, now the simulation is a little bit slow because we are doing calculations during the simulation. At each Monte Carlo step, there are those loops making that average sums, and that takes time. See, the blue line is growing to infinite. the pressure over the cells is, the average pressure is growing higher and higher accordingly with the growth of the target volume.
Gilberto, I'm sorry to interrupt, but we are at 4:20.
Okay, if 5 minutes sound okay, just to
Okay.
finish it. Okay, so the last thing we can do is try to control this growth. This is the curve you have shown, we've seen. And the control is with this, putting this function in the growth rate. This function will control the growth. Let us do this very simple, see? In a very simple way here, I will go to the Steppable, where I have this growth rate here. Now it's not in line 46, but since I messed with number line here, I will create here a reference, a pressure of reference, P high. Its value is 35, for example, okay? And when I do the loop, the cell target volume will be updated with this function here, like that. So is the max value of 0 and P half minus cell ontogenic pressure close. So this function will behave like that. And see, if P ref minus the cell pressure is negative, that means that the cell pressure is much bigger than the P half, then the cell doesn't grow, because we will have 0 from this function here. The only way to target increase is when the cell pressure is below the reference value. Okay? So save it. And let's start here. We still, at the beginning is the same, because every pressure is below that reference. What happens, the average pressure starts to reach a constant value. It doesn't grow anymore. And in average, see, as every cell has pressure above the limit, the reference value, there is no growth in the target value anymore. Everything got controlled that way. Okay? I think that's it. The other things you could do, it's inside an exercise in the students' materials. You can check it. 233 is to replace the function there, the function that I just added here to control the pressure, the growth, for another functions like that, for one of those, and see what happens. Okay, like an exercise for it. That's it then. Well, thank you.
We will launch the poll here in just 1 second.
but
All right. Thank you, Gilberto. So everybody, please do fill out the poll. We'll have a little bit of time for Q&A, and then we'll take a short break and move on to our fourth module for the day. So please, any questions that you may have, go ahead and feel free to ask now.
okay, stop sharing.
Was there a question?
Yeah, I have a question for Gilberto. Hi, Gilberto. It was a nice lecture. Really enjoyed it. So I had a question. Suppose I have a chain of cells, rod-shaped cells, linked together with cell linkages, and I want to grow it in a specific direction, let's say along the x-axis. So the initial configuration of the cell linkages is along the x-axis. So I want to grow it along the x-axis. So how do I specify that kind of direction in the steppable file?
Well, I don't think you can specify a growth in one direction.
So is it possible that the function g that you plugged in, can it take the form of a tensor, for example, where like e to the rt, suppose I want exponential growth with a growth rate of r and t is time. So can
Yeah.
it take the form of a diagonal tensor like e to the rt 1, 1?
I don't think you can, because when we say that the target increase, for example, it says to the cell, okay, you can grow anywhere. There is no preferential direction to the growth. It's a stochastic.
Okay.
You cannot say to the cell grow in that unless you use some external field, for example,
Okay.
to say if you find that field in that direction, grow in direction of the great, like chemotaxis, for example.
Okay.
You put something acting over the cells that will polarize, for example, the cell to grow in some direction. But not only from internal processes. You have to include something else in your code.
Okay.
This is the way we model the cell crawling for single cell crawling. Me, Pedro, and James did this. We have a field that we say that the cell produces something underneath. We have a cell with three compartments, three parts.
Mhm.
One of these parts is the lamellipodiums. We say that lamellipodiums secrete something, and this secretion favors the growth of lamellipodium. So
Okay.
we are saying the cell grow in that direction and move in that direction.
Okay. Okay.
Okay,
So
then you
there
can,
should be some kind of like a potential field or chemical
yeah,
potential.
something, yeah. But stochastically, you cannot do that. So you only
Yeah.
say growth.
Okay. Okay.
Huh?
I see. Thank you.
About this example that you just said, do you have a published work or anything that I could take a look at that? It just sounds a lot interesting.
For the cell crawling?
For the cell crawling, yeah.
Yes, I can. Pedro, do you have it in hand there? Can you put in
Yeah.
the?
I can even show how it works. I have videos, etc.
Yeah,
And
please.
well, kind of relating that question to what I work with, and I would believe that it's possible as well to somehow put a chemical gradient inside the cell instead of something that the cell responds to in an exterior, but maybe like how the proteins or intracellular mechanisms would work. I don't know if you can do it instead of an external chemical gradient and following chemotaxis with that. Instead, the protrusion comes from like a chemical field inside of the cell. In this case, it would be from RAC. I don't know if that helps. Fields inside cell. Okay, that's good. Thanks, James.
Yeah, this is it. And there's also this one in 2D. So basically, actually let me do this. I will show you guys two interesting simulations about that.
Pedro, maybe we better do that after 6 so that Peter can present
Okay.
his